cool. Morning, everyone. My name is Yi-Ting, and I will be presenting on our DNA-based monitoring in Saldanha Bay. This project was actually led by Dr. Robin Payne, who could not be here today because she's actually in the field. I was there with her earlier this week. We were diving, but more on that later. So I am a marine consultant and marine biologist at Anchor Environmental, which is a group of companies that offer a wide range of services, and we collaborate with many different companies. This includes City of Cape Town, Sandparks, Sea Harvest, Deb Marine Namibia. So in other words, we aren't actually just South African. We do have a Namibian office. We've worked with the WWF and the Department of Science, Technology and Innovation as well. Our work covers 3 thematic areas that straddle the marine, estuarine, freshwater and terrestrial realms. These themes are ecological research, monitoring and assessment, fisheries and aquaculture, and conservation and sustainable development. I'm not going to go into too much detail on all 3 sectors, but if you want to, you're most welcome to visit our website or just Google us and then hop onto our website. So very briefly, our ecological research, monitoring, and assessment work branches into 7 different categories. Is that? Oh, it is. These are ecosystem delineation, monitoring and health assessments, environmental flow assessment and water resources classification, biodiversity assessments, conservation and management plans, environmental and social impact and strategic environmental assessments. This is often for companies that are looking for offsets or marine protected areas. It's just checking legislation with what we have. Meteorology and oceanography, invasive alien and problem species studies, as well as water quality, waste management, and toxicology studies. For more detail, again, website. We have a blurb underneath all of this, and some projects are listed that we're allowed to share because other projects are private. So the Saldanha Bay DNA-based monitoring for biodiversity and the detection of alien species actually falls under 2 of these categories, because those 7 categories often, there's always overlap between them. And for this project, it actually is ecosystem delineation, monitoring and health assessment with invasive alien and problem species studies. Let's get into how we use the DNA and metabarcoding for these environments. Part of the wind. Oh, I was so confused there. So simply, DNA is a double-stranded helix found in all organisms. I know there could be discussion that it's not in all organisms because there's RNA viruses and such, but it's found everywhere, right, in living organisms, and we use it to identify species and whatever we're interested in. Except with DNA, there is an exception of where it's found. DNA is often classified like, oh, there's DNA in our bodies, which is mitochondrial DNA or nuclear DNA, but with environmental DNA, it's a collective of. We often collect it from water and sediment samples, which means, like everyone else has said, it's a very non-invasive sampling method because all organisms shed DNA. Like Gwyneth has said, from our skin cells, our mucus, our feces and hair, even just us sitting and standing here, we're shedding everywhere. So in the eDNA, the DNA is free floating and they're degraded fragments, and it's a great way to capture a snapshot of what was and is currently in the area. But with advantages, there are often limitations, but again, we'll discuss this later. We use eDNA metabarcoding to monitor the biodiversity, including the detection and tracking of non-native or alien species in Saldanha Bay. We do often work in Saldanha Bay. It's one of our main dive areas and sampling areas. And in that photo there is underneath one of the mussel rafts in Saldanha Bay, which we do survey and collect data from. So metabarcoding is a quick and cost-effective way of identifying species by selective gene regions and our focus was in 3 major harbors along the major shipping lanes, because Saldanha Bay is actually a major port for many, many different ships. Sample-wise, we use ballast water, which is water discharged from huge vessels, seawater, sediment, and wall scrapes that we use to send off. So when I say wall scrapes, I do just need to caveat this, because I learned on Tuesday, it's not actually always a wall, it's just a hard substratum that we scrape off of. So our sampling sites, this is Saldanha Bay, that's the Longabon Lagoon, and we classify this as North Bay or Outer Bay. This is Small Bay, and that is Big Bay. And this obviously is your lagoon, and on your, would it be easier if no, okay. These are our water and sediment sites. So often what happens is, Anchor's a dive company, so we have a group of certified commercial divers, and we often just go to those locations with our boat and we drop down and collect sediment and water samples. So over here is a commercial port that it's not very well known. It's a very small port, but it's still widely used by commercial companies. Pepper Bay Yacht Club is over there, and I'm sure everyone knows about Mykonos in Longabon. Their yacht club is there. So water sediments around that area, because we're capturing what is in that space. And then on the right is where our wall scrapes were located. As you can see, there's a wall scrape at the harbour, and another one there, and then one at Mykonos there. So we try to get as much of a picture around that area as we can, because we are limited by time. So as an interesting thing, if anyone wants more details on this area, these locations are actually coinciding with the Saldanha Bay Water Quality Trust, because we do benthic long-term monitoring for them, and every year we publish a report for them, which is publicly available. And if you ever want to explore anything with regards to marine oceanography, we have everything written, all the chapters. So sampling. As I said, we have a dive team. So we are registered as a dive company, so we often collect our own samples. The only sample that we did not collect is the ballast water that was provided to us by our client. And also cannot imagine climbing onto a vessel and going, "Can I please have some water from your tanks?" So we often collect using scuba, and so with wall scrape samples, these are all of these are examples except for my manager, who is floating on the water. As you can see, sometimes it's a wall, other times it's not, and sometimes it's just reef. Basically, we take a 25 by 25 centimeter quadrat, and we go down, we put it on a spot, and we scrape as much of that as out, so we can get as much diversity as we can in that area. It's usually, maybe not in these conditions, the person on top would sample at about 0.5 meters, so 50 centimeters below the surface. Usually we can do that on snorkel. I did that on snorkel this past week. It was a bit rough, I'm not gonna lie. And there's usually someone, I don't know if you can see that bit, but there's someone actually there at the bottom at 5 meters depth. So instead of buckets now we use hand nets because it's safer. Imagine just watching your sample fly away with the current. So there's that water sampling. We just, as we call it, we bomb down. Now those drop down really, really quickly. And then we take our bottles and flush it out and then fill it up with bottom water as well as surface water. So unfortunately, I do not have photos of our sediment samples. Same concept though. We drop to the bottom, collect the sediment, quickly close that jar so it's nice and full, and then come back up. So there's as glamorous as that all sounded, there is also field processing, which one would think if you collected the samples, you can just hand it over, but unfortunately not. So our wall scrape samples, we combine the surface and the bottom into a bucket, and then because we use eDNA kits from a UK company called Detectionof, they have special instructions on how to handle their technically our samples for them to sequence. And so we homogenize this, and then we subsample them. With the water sample, though, this is our makeshift peristaltic pump for in-field use. And we filter that water into a 0.8 micron filter. We preserve it, and then we freeze that as well, and then we send it off. So with this wall scrape samples and the sediment samples, obviously there will be a lot left over, and we preserve that in ethanol so that the labs at Anchor, we have a taxonomic lab, and it's morphological, it's not genetic. And we preserve these so our taxonomists can ID all the organisms in those samples. The reason why we preserve it in ethanol as well after IDing, we tend to send it off to an institution like the Africa Center of DNA Barcoding to get sequences barcoded. And there's much reason for that. So simply, what is DNA barcoding? You find something, an organism you're curious about, and then you take a tissue sample or a swab, and you extract that DNA, you split it up into barcode fragments, which get amplified, so copied over 1,000,005 times. It's not actually 1,000,005, I just use that as an expression, using a set of primers, and then you sequence the barcode. And then it comes up like little squiggles everywhere, which you stitch together. Well, usually our service provider would stitch it for us. I remember doing that in masters. It was not easy, to say the least. And then you take the barcodes that were generated and you put it into a database like NCBI, GenBank, or GBIF, and which are these databases with thousands and thousands of sequences, which you can match yours to theirs. And that's how you kind of identify what you find, right? Sometimes you won't get a species match, or sometimes you will get a species match that could be wrong. And that's how we go about finding what is in our sample. So Detectionof eDNA meta-barcoding kits targets the 16S region, which is mostly bacteria and archaea, 18S, which are mostly eukaryotes, and then your CO1 region, which targets your animals. So for the results of the Saldanha eDNA monitoring program, the ballast water used bacteria, eukaryote, and vertebrate pipeline. So a pipeline is basically their method of how to find what you want to study, right? But you can't have all of it together because it'll get lost in the mix. So they separate in different pipelines, and in the ballast water for bacteria, 9 phyla were ascribed with a maximum of 5 taxa per sample. Proteobacteria dominates the bacterial community, which is actually quite expected because they're a diverse and abundant microbial group that drives carbon cycling in the deep ocean. However, no marine bacteria are considered non-native or alien in South Africa because, again, it's a very understudied area, as everyone else has said. So, and with the eukaryote chart, 5 kingdoms were ascribed to about 17 phyla and 66 taxa. 8 of those were detected to species level, and of them, none were actually considered non-native or alien because they have been observed in the area by people. So with the seawater, these are a selection of species that were found using the vertebrate pipeline. This includes the American flamingo, the broad-nosed seven-gill shark, which is your B, the Cape elephant fish, your yellowtail kingfish, your Cape fur seals, and your southern mullet. Again, just a very small snapshot. It's actually an enormous table in our actual report. So in total, 44 taxa were detected, ascribed to 5 different classes, which were 5 unique birds, 37 fish, and 2 mammals. 26 taxa were identified to species level, and 8 matched reference sequences from databases like NCBI and BOLD, which is Barcode of Life database, I believe. Again, the majority of these species that were detected have been observed in Saldanha and South Africa, so they weren't considered alien. With invertebrates, I made a slide on its own because, again, Anchor has our own invertebrate taxonomy lab, so we take it very seriously. So with the eDNA barcoding, what was detected in the sediment were 23 taxa assigned to 7 different phyla, and the benthic taxa actually dominated the composition, specifically Cnidaria and Mollusca. Your Cnidaria are often like your jellyfish, or more squishy kind of, and anemones, the squishy kind of things that you find in the ocean. Your Mollusca are kind of like your sea snails and those hard-shelled spiral species. An invasive mussel was detected, and 2 gastropods that could be considered alien, but these were detected in single instances, which means they were only detected once in all of those samples, which we obviously need confirmation, so hopefully at some point we find actual samples of them when we go diving and sampling. For our wall scrapes, we used it as a proxy for fouling communities because, again, we scraped it off surfaces that often interact with vessels. These were dominated by mollusks, porifera, which are your sponges, and if Robin was here, she'd be very happy that you celebrate about sponges. The order Mytilida, which is part of Mollusca, was actually very dominant in that phyla. Yeah, Mollusca. Mytilida. Yeah. Like the sediment samples, there were single incidences where 30 were detected, 10 of which were alien. I'll show a picture later on some of them. And yeah, so wall scrapes, lots and lots of alien species. Sediment, not so much. And that could be because of substratum, the interaction between vessels and the environment. So, the detection of non-native species. The DNA-based survey with conventional morphological taxonomy identified 6 alien species. So, using those 2 methods, we found 6. Whereas with DNA, we found 18 alien, of which 14 were invasive. But with conventional morphology, we actually identified 7. So, according to these results, the greatest numbers of alien species documented were actually at the mouth of Mykonos Marina, which in the previous slide I showed is where one of the smaller harbors were, which is actually what we expected because there have been many accounts of high numbers of alien fouling species in the Western Cape within these small yacht basins. So these are all examples of the kind of, right, I have to use my finger, alien species that we found. This is a spionid polychaete. That is the Japanese skeleton shrimp. That's the porcelain crab. These are invasive barnacles. That's a Ballasidian. They actually look very pretty in real life. If anyone ever goes to Saldanha and goes to like Mykonos, you can walk onto the shipping, well, where they dock. Or at least I walked on there without permission. And my boss and I kind of hovered over the harbour, the dock walks basically, and it was covered in Ballasidians. It was bad, but equally so pretty. These are sea vases, and these are some of your invasive mussels. This is another invasive species that was detected by the wall scrape, which is the pink-mouthed hydroid, also part of the Cnidaria family. So, with the invertebrates, I showed you guys our results from eDNA metabarcoding, but now let's move on to the morphology of things. If we look at it, you can see there's quite a bit of discrepancy if you... with the eDNA, there were 7 phyla. With morphology, there are 5. And with wall scrapes, I mentioned 9 on that side, and as you can tell, so much more on this side. I'm so sorry for hitting your screen. So for example, Cnidarias dominated in eDNA barcoding, but Annelida, which are your segmented worms, dominated your morphology. And on wall scrapes, your mollusks dominated, whereas in eDNA barcoding and with wall scrapes, it was your arthropods, which are like your crustaceans, your shrimps, your hard-shelled organisms. What does that tell us? It makes us look at the bigger picture, right? How can you use only one method? Should you use another with another method? So eDNA is a great tool at providing a snapshot of a community and or the environment. It serves as an excellent baseline, so you know what's out there, right? But using eDNA has several limitations, especially in understudied environments and in the Southern Hemisphere. I'm sure like whoever has worked on anything in the Southern Hemisphere, looking for literature is a nightmare. So with eDNA, hydrological conditions are very important. In other words, your currents, your winds, if something gets taken from somewhere way far out into what you're sampling, you're going to find it there and you're going to go, oh my gosh, I found a shark here. Which, interestingly enough, a different project we had, we collected water samples offshore, South Africa and Namibia. We found ostrich in those water samples. But that is also because it was close to relatively close to the Orange River mouth, and because of hydrological conditions, it got washed down. Ostrich, middle of the ocean, I really don't think so. And so that also brings limited literature and existing databases. I've spoken about comparing the sequences you find to NCBI and BOLD and GBIF, but it's only your eDNA sample will only look as good as what is available. Because you can always blast something and something may not come up, or you only match potentially like an order or a superfamily and not really get to the nitty-gritty bits. And we have also had our sequences matched to a completely different species from a whole different phyla in Europe. It's one of those things where it's very tricky, especially when databases like these aren't regulated, because you're welcome to just upload your sequences. And again, with eDNA and our understudied marine environments, it's such a dynamic environment that with your single incidences, the actual animal could be there, and this is why we may or may not struggle finding our second records of certain invasive species. Traditional methods are difficult to use, because if I'm grabbing a sample here and the alien crab is sitting here, how will I know it's actually there by using morphology, but it's shown up in eDNA. It's always good to have different methods to confirm your findings, because you can't just say something is what it is without support, as we've always learned in science. So for this project, combining traditional and DNA-based methods aided in documenting more alien species than any of our previous surveys in Saldanha. And we have been monitoring Saldanha for, I want to say, a decade. Yeah. Again, that report, publicly available on our website. So for our take-home messages, what I want to highlight is that eDNA and conventional morphology was used to detect alien species and to assess the general biodiversity of the environment. Together, we identified 31 invertebrates, with eDNA identifying 18 unique invertebrates, conventional morphology 7, and together we found 6. All the barcodes that we have ever generated, we do our best as scientists to upload and bridge gaps in the global databases, because it's huge. Sequencing a marine worm, we have lots of actual side projects at my company. Getting that to match to anything nearby was 0. We could not find anything. Even with my masters, with my sea cucumber, I could not match anything close by. So at Anchor, we do try our best to bridge any kind of gap in the scientific realm when we're allowed to share data. And yeah, so I think our sequences are on BOLD and GBIF. Thank you guys for having me. And I do just want to put out there, especially for your students, we do offer internships. We often have job shadows come into our office to work with us. Usually what happens is we'll have, at Anchor, we have different teams. And I'm part of the benthic team. We have like a fisheries team. And so usually we'll get whoever comes in to be involved all over, and if they're interested in something, we'll always try to make a plan to have them speak to someone that has that knowledge. We're based in Tukai, and if you want to email us, there's the email address, and our HR will direct you to the correct person.